Hello everybody and welcome to our EEG-fMRI webinar series today. I'm Tracy Warbrick and I'll be leading you through the first session, which will be an introduction to EEG-fMRI. So just before we get started with the content, I would just like to introduce you to a few features of GoToWebinar, just in case you've never used it before. So your interface will look like this. Hopefully you're not seeing foundation slides, but you're seeing us. And what you will notice is that you're muted. So all of our attendees are in listen-only mode, and this will stay that way throughout the webinar. This is for our speakers' comfort, but also to protect your privacy, so we can't accidentally listen in to what you're doing in your labs today. If you can't hear me, you probably can't hear me talking about audio settings, but hopefully you can see this. And you can change your audio output in the settings tab, so please go there. Now, we do have a handout in today's webinar, and this contains a list of resources that our speakers will be referring to throughout the webinar. You can download that in the handouts pane, where you can find that here, highlighted with the yellow circle. We do have a reactions button in GoToWebinar, and we're quite happy for you to use that. We do like to get feedback from you guys. It's nice for us to know that there are still people out there listening, but please don't use the raise your hand option. We have quite a lot of people online, and it's not possible for us to take questions on the fly today. You can also customize your view a little bit. So if you're new to GoToWebinar and you prefer to focus on the slides or not have any videos or zoom in, then you can use these various controls to help you with that. And then we would also like this to be an interactive webinar. So it would be really great if you guys could ask some questions, and you can do that in the questions pane that you can see here. Now, we will then deal with the questions in one of three ways. So one of the workshop team will either reply to you directly within that pane. If we think this is a great question that everybody would benefit from the answer, we will ask it live during our Q&A. And if we have too many questions that we can't deal with today, we will reply to you via email. So all questions will be dealt with today. So don't worry if you don't get an immediate response, we will deal with it at some point today. Okay, so before we get into the content, I would just like to introduce your webinar team for today. So I've already introduced myself, I'm Tracy. Also online today we have Celia Yeager, and she is our application specialist for EEG-fMRI. And we have Michael Hofstetter, who is team leader for the scientific support, and he will be leading you through some analyzer analysis steps today. So between them, Celia and Michael will take care of the second two webinars, and you'll hear a lot more from them later. Just to give you an overview of what's happening today, we have the introduction to EEG-fMRI that I will do, and that will run until 2 p.m. CET. We will then take a 15-minute break, and then Celia and Michael will take you through webinar 2, which will be the data pre-processing and considerations. We will then take another 15-minute break. I'm sure we will all be needing some coffee by that point, and then we will restart again at 4 p.m. and we will have our final session, which will be combining the EEG and the fMRI data. And Celia and Michael will take you through some ways to do that. So you will hear more from Celia and Michael later, but for now, I will take you through an introduction to EEG-fMRI. So see you guys later. Great. So as I said, I'm going to do an introduction to EEG-fMRI, and this is for any of you that are quite new to this topic, and we can just give you some background as to why we might want to do it, any considerations that we need to take into account that will hopefully lay the foundations for the data analysis things that Celia and Michael will do with you later. So the first thing I'd like to think about is multimodal imaging in general. So generally, we're wanting to measure neuronal activity. So we have something going on in the brain and we want to measure that. And we can do that mostly by measuring difference potentials at the scalp. So on this side, we have an example of either using EEG for the difference potentials or the magnetic fields associated with those potentials that can be measured with MEG. We can also measure blood flow related or hemoglobin related measures. So we can look at blood flow with ultrasound, for example, or with fNIRS. And what we're really interested in today is looking at the fMRI side, which is also related to blood and also the hemoglobin content of that. Now, if we just think about EEG and fMRI's techniques, I'm sure most of you are familiar with at least one of these techniques, perhaps both of them already. And we always talk about the various resolutions that we have with different neuroimaging methods. So Our EEG data and also MEG has a very high temporal resolution, but it has a low spatial resolution. And our MRI, on the other hand, or our fMRI, has a high spatial resolution, but a lower temporal resolution. Now, it's worth bearing in mind that both of these things are non-invasive. Now, if we want to get to this space down here, where we have high temporal resolution and high spatial resolution, we can consider invasive options such as intracranial EEG. But in research applications, that's not so common. So sometimes they're used more in clinical applications, but not so much in research. So what we want to do is to try and get to this space where we have temporal and spatial resolution at an optimal level. And we can do that by combining EEG and fMRI. So here where we have these two measures, we have complementary information in terms of the spatial resolution and also the temporal resolution without having to use an invasive technology. So this is why we might want to combine our EEG and our fMRI data. So the next thing I would like to talk about is how we measure both of these signals and what the neural basis of those signals is. And the reason we want to cover this is to give you a flavor of what we are actually measuring, because we're going to talk about data integration techniques later, it's really important to understand what data you have, how it was acquired, and what kind of properties that data has in terms of its temporal or its spatial resolution, for example. And that will give us a better idea of how we can combine them and why the methods that Celia and Mikhail will show you are very relevant for this particular application. So if we first consider EEG, we have a number of components of the EEG system. So we need something to detect the signal. So we generally use an EEG cap for this. So for us, it's our brain cap MR. We then need to amplify the signal and convert it to a digital signal. And this is where we use an amplifier. So we generally use a differential amplifier for EEG, where we have a reference electrode and a signal electrode, and then an analog to digital converter. We then record the data. The reason I'm mentioning this is that there are some recording parameters that are very important to acquiring the EEG data inside the MRI scanner. We do have some additional resources on topics such as how to set up Recorder, and so I would encourage you to take a look at the handout and follow the links on there to find out how to set that up appropriately for EEG-fMRI. But because it is a special application, there are some special things you need to take care of in terms of setting up your workspace. And unfortunately, when we record the data, it doesn't come to us in an automatically usable state. We have to do something with it. And so we have to pre-process the data, or we have to handle the scanner artifacts. And then we have to analyze the signal of interest and potentially export that. And those are the things that you're going to learn about a little bit later today. So those are the components of our EEG system, and we do need to consider how they might interact with our MRI system to understand a little bit about what's going on with the data. So we will come to the MRI system in a little while, but first I would just like to look at what we're actually measuring with this EEG system that we put inside the scanner. So I'm just going to give a very brief overview here. So generally what we will have is a stimulus that's presented to our participants, or a change in state, so something endogenous to our participant. And this will result in a population of neurons firing. Now, if we have synchronized neural activity, so a bunch of neurons firing at the same time in response to the same stimulus, we do generate a potential that can be measured with our EEG electrodes at the scalp. Now, this is great because EEG is on a millisecond time scale. So by measuring these neural signals with these electrodes at the scalp, we can access the rapidly changing neural dynamics that are associated with the processes under investigation. However, this does have some disadvantages. So as you can see, our signal generated here is measured here. And it has to pass through all of these different tissue types in order to be able to get to where we want to measure it. So it arrives at the scalp in an attenuated form because it has to pass through all of these different tissues, but also we're not always exactly sure where we're measuring that signal from. So just because this electrode is directly above this synchronous neural activity, it doesn't mean that that's the only thing that electrode is picking up. Maybe there's something going on here, or maybe we have another electrode here that is also picking up contributions to that. So with EEG, it's not always easy to tell where the signal is being generated from, even though we have a very good temporal a temporal resolution with EEG. So for those of you who are not so familiar with EEG, who are maybe coming more from the MRI side, what do we actually measure with EEG, or what are the traditional types of signals that we look at? Well, we can broadly divide them into spontaneous activity. So this would be ongoing background activity in the EEG, and we can look at the power of different rhythms within that. So there are different frequencies of activity that we can look at. A common example is alpha activity, that you would expect to see an increase in alpha activity in the occipital regions if you close your eyes. So we can look at the power of that particular rhythm in response to doing something like closing your eyes, or we can monitor the background fluctuations of those things. The general other way we can look at EEG is a more task-related way of doing an experiment. So this is where we would present a stimulus at time 0 here, and then we would look at the response averaged over time. Now you'll notice here that I have a time scale of up to 500 milliseconds. So what you will see is that within that 500 milliseconds, we can actually differentiate a few different sensory and cognitive components. So an event-related potential has a number of positive and negative going peaks, and these generally reflect well-defined sensory or cognitive components. So we can look at early sensory components, for example, with the P1 or the N1. And we can also tap into later cognitive components, such as making a decision about two different stimuli, or the way that we process those stimuli. So for example, the P3 that is elicited if you see a rare or a target stimulus compared to frequent ones. So in general, our EEG can be measured in terms of the spontaneous background activity, or the power in different rhythms of the EEG, or in event-related potentials. Now, this is just a broad categorization of two ways of looking at the data. There are, of course, many ways that you can analyze EEG data. Okay. So that's a very brief overview of what EEG is, what we need to measure it. So now let's do the same for fMRI. So in terms of what MRI is doing, we're essentially relying on the fact that hydrogen nuclei emit some energy if you stimulate them, and then they return to baseline. So the MRI measurement is based on this energy that's emitted from these hydrogen nuclei. Now, the energy emitted varies across the different tissue types, and this is how we can look at different types of tissue, for example, grey matter, white matter, or even blood oxygen, for example, by using MRI. So what we have in terms of the MRI, so for our EEG system, we have our electrodes, we have our amplifier, and so on. In terms of the MRI, we have a number of components that are vital to making the signal, but also interact with our EEG system, which we will come on to in the next section as well. So the main components are the magnet itself. So this is the static field. So for example, you may have heard of 3 Tesla or 7 Tesla magnets. That is the field strength. And the purpose of this is to align the protons when we're inside the scanner. We then have an RF coil. So this is RF radio frequencies, and this is used to excite the protons. And then we measure the energy emitted from those as they return to equilibrium. Now the gradient fields, so this is what you would hear as acoustic noise in the scanner. So the gradient fields change across time and spatially, and this allows us to select a specific slice of the brain in terms of what we're going to measure. So we don't necessarily measure the entire brain in one go. We tend to measure it in slices, and these gradients allow us to do that. So that's essentially the basic components of an MRI scanner. As with the EEG, the MRI scanner doesn't automatically give us usable data that will answer our research question. We need to go through some other processes like image reconstruction, pre-processing, and also statistical analysis and modeling. And the analysis and modeling part and some of the pre-processing are things that Celia will go through with you later. So that's essentially what MRI is doing. And remember, we have our EEG system that we then put inside of the MRI scanner. So keep that in mind when we come back to the interactions in a few slides' time. So now let's think about the nature of the MRI signal. So I've already said that they're quite different, the EEG and the fMRI, in terms of their resolution and so on. And if we just take this flow diagram as an example, we have our stimulus and our neuronal activity, and we would measure our EEG here at this time point. But with the MRI, we have this whole process of other things going on before we actually measure the MRI response in the scanner. So this part of the figure is quite busy, I'm aware of that, but this is for good reason, to show you that this is quite a complex process. So if we do have some excitation or synchronous neural firing, then this increases the metabolic demand. And if we have an increase in metabolic demand, then we need to get some additional energy to that area. And this happens by changes in blood flow. So when we have the stimulus or the change in neuronal activity, we have this process called neurovascular coupling. So this is where an increase in blood flow will happen to meet that metabolic demand of your firing neurons. Now, what also happens is oxygen is used by the areas that need it, and this is carried by hemoglobin. So what we use for fMRI is something called the blood oxygen level dependent imaging technique. And this takes advantage of the magnetic properties of hemoglobin, which differ when it's oxygenated or deoxygenated. And this is how we can then measure our BOLD response, or our brain's response to these neural activations. It's looking at this whole process of neurovascular coupling, metabolic demand, and the delivery of oxygenated blood to areas that need it. So the BOLD response itself, as you can probably see from the previous diagram, is a correlate of neural activity. It represents this complex process of neurovascular coupling. It's not a direct measure of the neuronal activity. So as a result of that, these changes in blood flow that we measure with BOLD imaging actually happen seconds after our event, our neural event. And consider that the image I showed you for the EEG event-related response was on the scale of 500 milliseconds. This one is on the scale of 25 seconds. So what you can see here is that if we have a stimulus at time 0, our peak BOLD response is likely to be 5 to 10 seconds later than that, because what we're measuring is this whole process, or this complicated process of neurovascular coupling. So this is why the MRI response is on a completely different scale. And this is something you need to consider when you're doing your analyses, and Celia will talk you through the differences in these temporal scales later when she's looking at how to analyze the data. So while the BOLD response is slower than anything we measure with the EEG, it does provide an excellent three-dimensional map of regional activity. So we do get a very good idea of where things are happening in the brain, which, as we said, for EEG, the spatial localization is not so good. So as I said towards the beginning of the talk, when we looked at that diagram of where the temporal and resolutions can be brought together to maximize the data that we're measuring, this is where the EEG and the fMRI become very complementary. So we can tap into the strengths of each of these measures in a way that maximizes the information that we get about the process under investigation. Again, this is dependent on how you analyze the data, and you will learn something about that in the later webinars. So in terms of common fMRI measures, we can look at the fMRI data in similar ways to the EEG. So we can look at spontaneous changes in the background activity, for example, resting state MRI. So initially, BOLD responses were measured in task-related activity, but advances in analysis techniques do mean that it's possible to measure intrinsic spontaneous activity as well. So for example, if we consider the brain as a network of connected regions and patterns, we can look at the connectivity of those patterns of activity and see how those change over time. What we will focus on with our data in these talks is a task-related response, so the event-related potentials in the EEG, and this would be looking for a task-related BOLD response. So typically for a task-related BOLD response, we're looking at a period of activation against a baseline, so it's a relative measure. So we would usually compare the baseline activity to the task, and this will give us a spatial map of the brain regions that are involved in that particular task, or that are active during that particular task. So it's a baseline versus task, and you will model that in your analyses, and Celia will talk a little bit about modeling your GLM later. Okay, so let's have a look at the combination of the data just to remind ourselves what we have here. So we have our neuronal activity firing here, and this is where we measure our EEG data. So we're measuring EEG here, then all of this happens and we measure our bold response. So just to take a look at these two task-related or event-related signals side by side, here we have our EEG again and our bold. So again, just to note that we have this one hemodynamic response in response to the stimulus with an initial dip, a peak, and a post-stimulus undershoot. The most common measure is to look at the peak or to at least look at the area here. Whereas in EEG, we have a very short time scale of 500 milliseconds, and we already have a number of components that we can look at. So in terms of the temporal resolution, this is the advantage of doing both of them. But as I said, you need to be aware of this, and also you need to understand how this influences your data analysis later. Okay, so let's just have a think about why measuring complementary data sets might be useful. So having these complementary data sets in response to the same changes in brain activity, whether that be spontaneous activity or evoked activity, is very valuable. So there are some cases where this is really the preferred option, so where separate sessions of EEG-fMRI, for example, just would not do the job. So for example, spontaneous resting state fluctuations, these are spontaneous by nature. You can't necessarily replicate that in separate sessions for EEG and fMRI. There are just different things going on. It's a different environment. Other endogenous activities, such as epileptic spikes, also benefit from simultaneous EEG-fMRI. So if you're measuring EEG in a separate session and you have some epileptic activity, you can't ask your patient to go into the scanner and replicate that epileptic activity. That just doesn't happen. It's spontaneous and endogenous activity. So measuring the two things at the same time is truly beneficial for applications such as epilepsy. It's also very useful in cognitive neuroscience experiments. So while it is possible to do the same experiment in the EEG and the MRI separately, you are then in different environments. So for example, in the scanner, you're lying down. In a lab, you may be sat in a comfortable chair in front of the screen. You may be viewing a visual task through goggles or a screen in the scanner, whereas you're probably sat in front of a high-resolution screen in the lab. So there are so many factors that may differ between the two different recording environments that measuring them together under the same conditions actually gives you a better indication of how these two signals co-vary. There are other advantages of simultaneous EEG-fMRI from a purely practical point of view as well. So it eliminates habituation effects. So if you're doing a task on multiple sessions that has some kind of learning effect associated with it, then this can affect your results. So while we do counterbalance our order of tasks, if we do have multiple runs of the same task to try and account for that in the group level effects, you will still have some individual level habituation effects, and that could influence your analyses in the end. As I mentioned, having the same sensory environment for all measures is practical, and it means your data are more comparable. And also reducing the overall time required for the experiment is a real practical consideration. It may cost you less to have less lab time or to have to pay your volunteers for a shorter time. And it also can help reduce attrition rates of volunteers as well. So sometimes if you have to do 2 sessions on different days, people don't always come back for the second one. So there are some purely practical aspects of doing combined EEG-fMRI. So while all of that sounds great, just because we have EEG-fMRI at our disposal, it doesn't mean we absolutely have to use it. So sometimes separate sessions are actually better. And so what I'm going to cover here on this slide is some things that you should think about when considering whether EEG-fMRI is for you. So the first is, what is your research question and what responses do you actually expect? So what brain activity or brain responses do you hypothesize for your study? And are they really likely to be present in both modalities when measured under the same conditions? You should also consider your experimental design. So is it possible to use the same task for both the EEG and the fMRI? So we've already looked at the different temporal scales of these data. So if you have a very rapid ERP experiment for EEG, for example, if you just use that exact same paradigm in the scanner, are you going to be able to measure bold responses to all of those stimuli as well? Or do you need to make some kind of adjustments? So you need to think about the task that you're using and how to design it. And again, you need to think about habituation and training effects. If you're measuring something that isn't susceptible to those effects, then maybe doing them at the same time is not absolutely necessary. So the other thing you really should think about is the data. And as I said, you're going to learn a lot more about the data handling later, but you should think about your data analysis in your strategy for integrating your data right at the beginning when you're planning your experiment. So do you intend to integrate the data sets or analyze them separately? If you're not planning a truly integrated analysis, then maybe doing them in separate sessions is not such a bad thing. And the other thing to think about is the artifacts. So are they acceptable for your planned analyses? I will cover a little bit about artifacts in the coming slides, but for example, maybe the EEG that you're interested in is at exactly the same frequency that you have some noise in your data. So you need to be consider how the environment is going to affect the data that you have and whether you can really get to your signal of interest if you measure it in the scanner. So as I said, EEG-fMRI is an excellent tool, but it's not always the right one. And you should make some smart decisions about whether this is the right tool for you. So let's just come to our interim summary here. So the pros of EEG-fMRI are that we have a complementary data set to the same stimuli under exactly the same conditions. And we hypothesize that we have the same neural activity contributing to both signals. So we're measuring responses to the same stimulus and the same neural activity with two different measures at the same time. And also the practical aspects of not needing two sessions are not to be ignored. Now, every method, and especially multimodal imaging method, does come with some disadvantages as well. So you have to be prepared for a decrease in signal quality in both modalities. So as I mentioned earlier, when I was explaining the components of the EEG system and the MRI system, they tend to interact. So what we get is artifacts in the EEG from the MR environment and also potential effects on the images from the electrode leads and so on inside the scanner. Now, also, EEG-fMRI can be technically challenging in terms of setting everything up and making it work. And we also have to consider safety aspects. So we want to have safe measurements. Now, even though I've listed a few cons here, so a few disadvantages of that, there are very easy ways that you can make this work and take advantage of the technique without worrying too much about these things. So what I want to do in the next section of the talk is describe how you can address these potential difficulties to make sure that you have successful EEG-fMRI experiments. Okay, so challenges and how to overcome them. Essentially, we want you to have successful EEG-fMRI measurements, and I'm pretty sure that's your goal as well. So what I'm going to do is cover those things that can help you get there. So we have safety, MR data quality, and EEG data quality. Now, all of these three things are related to interactions between the scanner and the EEG system, and I'm going to cover those in the next few slides. And the fourth thing you need to think about is data integration. I'm not going to cover that here because that will be covered in much more detail in the following two webinars. Okay, so let's have a think about safety. So this webinar series is about data analysis. I just want to cover a little bit about safety to show that it's not a problem that can't be overcome, but I'm not going to go into too much detail. But what I would do is recommend that you check out all of our other material on how to do safe EEG-fMRI experiments, and you can find links to those in the handout that I referred to earlier. So if you are doing EEG-fMRI or you're just starting out on your EEG-fMRI journey, it's a really good place to start. Is the safety aspects. So essentially we have an EEG system that contains electrically conductive materials, and we also have a conductive circuit. So if you remember earlier, I mentioned the differential amplifier that has a loop between the reference electrode, the recording electrode, the amplifier, and your volunteer. And what we're essentially doing is putting something that doesn't really like being in a magnetic environment inside a big magnet, and then running some radio frequencies and gradients as well. So the MRI environment is not a friendly place for such devices. So what I'm going to do is just cover a few of the risks and what you can do to mitigate it. So we've probably all seen these kind of images where something is stuck in a scanner that shouldn't be in the scanner. In this case, it's a chair. But the risks associated with a static magnetic field is that anything ferromagnetic will be displaced and drawn into the scanner. So you have to be very careful about what you take into the scanner room. The biggest risk that we are worried about is heating. So the radio frequency field and the switching gradients can induce electromotive forces, and these generate a voltage that can cause a current in a loop or in lead wires. And as we already mentioned, our EEG system is a recording circuit. It's an electrically conductive circuit. And there's also the potential for antenna effects where current can be induced along the length of a lead wire. Now, these two images here are showing worst case scenarios. So we have set the equipment up deliberately to try and make it get hot. So we don't recommend that you have your EEG cables curved like this. And this is also showing that the amplifiers can be susceptible to heating as well. And what we do have is a number of research resources, especially a support tip written by Celia quite recently that covers our safety updates and gives you some advice on how to prevent these things happening. So as I said, this is not a safety talk, but we strongly advise that you follow up on that if you're doing EEG-fMRI. So what can we do to solve these safety issues? So essentially, we can make sure that we don't use ferrous materials and components. So that's on us to design the equipment in a way that makes it safer to use in the environment. And we have our MR conditional equipment that can be used under certain circumstances with the MRI scanner. And we tend to use non-ferrous materials for that where possible. We also include current limiting resistors on the electrodes and connectors and lead wires, which helps to prevent heating along the lead wires. We can also consider using materials that are less conductive. So for example, on our long ECG leads that we have on our brain cap MR, we use carbon for the lead wire rather than copper because we have a longer wire there, and the carbon is less conductive. You should also consider the number of electrodes and the configuration of the cap. So the more material you put in there, the higher the chances are of something heating. So really consider whether you need such a high electrode count for your EEG-fMRI studies. From the MRI side, the most important thing is careful selection of the MRI sequences for EEG-fMRI. So some sequences are just not acceptable for EEG-fMRI experiments, such as those with a very high RF energy or those with inversion pulses that can cause issues for heating and damage within the equipment. Even the sequences that are acceptable for EEG-fMRI, such as your EPI sequences for bold imaging, it may be necessary to adjust your imaging parameters, for example, if you're using multiband MRI. So there may need to be some compromises on your sequences in order to make it a safe recording. And also consider the gradient performance mode of the scanner. So MRI scanners are getting stronger and more powerful, and they are coming with higher performance modes. And we strongly recommend that you stay in normal mode to try to prevent incidences of heating of the amplifiers. So as I said, safety is a very small part of this talk, and that's all I'm going to say on safety. And we recommend that you follow up on that. So let's come to our EEG data quality. So as with any kind of EEG recording, it's susceptible to artifacts. So those things that we will record anyway, even if we don't want them. So things like eye movements, muscle tension, those things will still be picked up inside the scanner. Electrical interference tends to be a little bit less problematic because the scanner rooms tend to be very well shielded. So you tend not to get general line noise like you would get in the lab, but it is possible to get electrical interference if you have other devices in the room. So for example, if you have medical monitoring devices or stimulation devices, then it is still possible to get electrical interference. We then have a number of artifacts that are specific to the MRI environment that do need some attention. And this comes back to our interaction of the EEG system and the MRI system. So this is why it's important to understand what they're both made of and what they're doing. So The general mechanism that induces artifacts in our EEG data is electromagnetic induction. So we really just need to go back to our school physics days and learn about Faraday's law of electromagnetic induction to understand why we have artifacts in the EEG data when we record it in a scanner. So essentially, a magnetic field will interact with an electrical circuit to produce this electromotive voltage. Now, as we already mentioned, we have a differential amplifier. All of our EEG electrodes are recorded relative to our reference. We have our participant in between, and we have the amplifier. So what we have here is a conductive circuit. We then put that inside the magnet, and so we are putting it in an environment where we are likely to induce electromagnetic induction. So any change in this magnetic field will induce a current in those electrode leads and will generate voltage potentials. Now, this change in the magnetic field can come from the magnetic field itself changing. So I talked a little bit earlier about gradient switching. Those gradient fields that we need to be able to select our slices, that is a magnetic field that changes in time and space. So that will induce artifacts in the data. And we can also induce artifacts by moving the equipment relative to those magnetic fields. So the magnetic field itself doesn't necessarily need to move, but if the equipment moves relative to that, then we can induce voltages there as well. So the common artifacts that we would see that are specific to being in the MR environment are the gradient artifacts. So as I mentioned, these are due to the gradient switching. And these are very, very large in amplitude, and they are also high frequency. So really, if you don't deal with these artifacts, you don't see anything meaningful in your EEG data. So it's essential to handle those. Then we have cardioballistic artifact. Now, this is a special kind of motion-related artifact, and it's essentially due to motion associated with the cardiac cycle. So pulsing, blood flow, any movements, for example, in the blood vessels, the pulsing of the blood vessels underneath the electrodes, and it's all related to how the equipment moves in relation to the field. And these are tiny, tiny movements. So if we're talking about potential pulsatile effects in the temporal regions, we wouldn't see that with the naked eye, but we do see that inside the scanner because the magnetic field induces these effects. And then we have motion-related artifacts, or general motion-related artifacts. These are things such as spontaneous head movement. So even if we try and keep very still inside the scanner, and that's also beneficial for our MRI data, so being still is generally a good thing in the scanner, we can't completely prevent spontaneous movement. So we do sometimes see head movement in the EEG. And another source of motion-related artifacts is specific to vibrations. So the movement associated with vibrations can also look a little bit bigger inside the scanner than outside. Now here I've just given you an overview of what kind of artifacts you can expect. In Celia's talk, she will show you what these look like and what it looks like when you've corrected them. But what we won't do today is process the data in that way. So we won't do the scanner artifact related rejections today. We will start from a cleaned data set. So if you're curious about how to do this in Analyzer, or if you're curious about what these artifacts look like in a little bit more detail, then we recommend you check out our webinar on scanner related artifacts for a little bit more information. So in terms of the solutions for handling these artifacts, we do have the artifact handling procedures, such as average artifact subtraction or carbon wire loop regression. And we do have a webinar specifically on carbon wire loop regression, if that's interesting for you. And then there's also practical things we can do, like designing the cap. So you will see that our leads tend to get shorter as we develop the caps. The shorter leads are less susceptible to vibration artifacts and also to some other artifacts like gradient artifact. We can add sensors for motion detection, so things like the carbon wire loops or a reference layer that we can then use to regress that signal out of the data. And there's also a number of practical things you can do. So the position of the equipment and the cables with respect to the z-axis of the scanner will influence, for example, the amplitude of your gradient artifact. Securing the cables properly can reduce your vibration artifact. so you can help yourself out a lot by making sure that everything is secure. And you can also reduce head movement by securing the head a little bit with cushions, for example. And we do also have a webinar on setting up an EEG-fMRI experiment, where we do cover a lot of the practical aspects of getting a good recording. And again, the link for that is in your handout. Okay, so where are we? We are on MRI data quality. Okay, so as I said, the systems interact. So if we expect an effect on the EEG data by being in the MRI data, we can, by being in the MRI scanner, we can also expect an effect of the EEG on our MRI. Now, when we're doing MRI, we want our magnetic fields to be homogeneous. And anything that we put into that field can affect the homogeneity of those magnetic fields. And this will affect your image quality. So if we think about the static magnetic field, so this is the field strength of the magnet, it's always there. And if you put anything into that field, it can influence the homogeneity of it. So what we're talking about here is susceptibility artifact. And susceptibility is the property of a particular tissue or a particular material to be magnetized when it goes into a magnetic field. And our EEG system has different magnetic susceptibility properties than human tissue that the scanner is designed to image. So by putting our EEG equipment into the static field, we can influence that field and our images by introducing susceptibility artifacts. If we also think of the radio frequencies, or our B1 field, we also want this field to be homogeneous as well. And this field can be affected by conductive materials. And as we already mentioned, our EEG system is a conductive material. So if we put an EEG system inside the scanner and we're running the radio frequencies and we have a B1 field, we can introduce inhomogeneities, and this will result in intensity variations in the image. So I'm just going to show you a couple of examples of these. So the first I'm going to look at is susceptibility artifact. Now these are quite extreme ones, and you can find loads of these kind of images on these websites. This one is for dental braces. So we lose most of the face here, and this one we have a hairband with a metal part. And this one is quite interesting because you see it's close to the head, and so we do get a huge signal dropout, meaning that we don't get any meaningful data underneath where that piece of metal is. Now you can imagine that if we put an EEG cap in there, we could have similar effects. Well, the good news is our EEG caps don't have such dramatic effects, and they're designed in a way to make sure that that doesn't happen. And what you can see here on the left is our RNet MR, and on the right, our brain cap MR. So if I just start with the brain cap here, what you can hopefully see is some very small indentations along the scalp. You can't actually see the electrodes themselves, but you can see a little bit of signal dropout underneath them. But it's nowhere near as dramatic as what we see for the hairband, for example. So the important thing to note here is that the EEG cap does influence your image, but it doesn't influence the brain data. So you can still analyze your data appropriately. The only thing you should really be concerned about or keep in mind is if you're going to do segmentation of your imaging data, that this may influence the segmentation of the skull area and those layers around there. For the RNet MR, it may look like this is worse, but it's not. It's just that you can see the electrodes themselves. So they're made of a different material that actually shows up on the image. So these are the electrodes in our RNet MR, which is a sponge-based system. So they're a little bit bigger than the brain cap, so that's why you can see them. But what you will see is the susceptibility artifact is the same. It's just a tiny little indentation in the scalp. So the point here is that you should be aware that your EEG system can influence your image, and you should look at it, but it shouldn't affect your data analysis. Now, the other thing we mentioned is disturbances in the B1 field. So what we have here is some simulated data that shows what a B1 map would look like with no EEG cap. So we have some difference in the signal intensity in the middle of the head here, but generally around the head, it's fairly homogeneous. What happens if we put wires, electrodes, or resistors in there, or even a full EEG cap, is that you can see we get some disturbances to this pattern, and it's no longer uniform. And these differences or inconsistencies in the field will influence your data quality. So what we also see here is some real data with no EEG cap and an EEG cap. And what you see with the 64 EEG cap is that we have a drop in signal intensity here. So the signal to noise ratio is going to be affected in this image. And this is due to the cable bundle that would be at the top of this particular cap. It's worth noting that this was during a development project, so the final version won't look as dramatic as this, but this is a nice example of how having a lot of material can really influence this B1 field. Okay, so what can we do about this? So the severity of all of these effects depends on a number of things. So it depends on the magnetic susceptibility of the materials used. It depends on the conductivity of those materials. Also the spatial orientation, particularly the susceptibility artifacts in the B1 field. You won't always see every electrode on the scan. It really depends on the orientation with respect to the imaging fields. The field strength and the number of electrodes. So what should we consider? Well, from our side, we have to consider the materials used, and we do extensive research and development to use the right materials for our electrodes, lead wires, and resistors. We also have to consider how the caps are constructed, so try to avoid having big bunches of cables or lots of electrodes in the same place. And this is also something to keep in mind if you have a choice between a number of electrodes in your system. So if you have a choice between 64 and 128 channels, for example, having 128 channels in there is likely to affect your image quality more than 64. So you should really consider what is appropriate for your data analysis. Again, thinking about what you want to do with your data while you're planning and designing your experiment, because it will have an influence on what you measure. So the take-home message really for this part is that you should be aware of how your setup affects your imaging data and then know how to deal with it later. Okay, so just very briefly, I said I'm not going to talk about data integration too much because Celia and Mikhail will do that. But essentially, we've solved the safety, practical, and data quality issues. But now what we want to do is integrate the data. So essentially, we have three ways of integrating the EEG and fMRI data. One of them is comparative. So we would take a measure from each, or a number of measures from each of them, and see which ones yield the most significant correlations between the two. So we're correlating different methods from one measure and another. Then we have symmetrical approaches, which integrate both modalities into a joint analysis. So for example, a joint ICA analysis. Then we have our asymmetrical approaches, where information from one modality is used to predict or constrain the other. Now, these asymmetrical approaches have been used much more frequently than any of the others, and we can look at it from an EEG-informed MRI perspective or an fMRI-informed EEG perspective. And Celia and Mikhail will take you through an example of each of those later, so we will be focusing on the asymmetrical approaches. Okay, so again, an interim summary before we move on to the final part of my session. There are challenges to successfully recording meaningful EEG-fMRI, but the significant developments in the hardware and the analysis technique in the last 20 years has pushed this technique forward to the point where it is a mature, established method. So the goal of a successful multimodal imaging setup is to make sure that all measurements are performed safely and that the data obtained from both measures can be optimized. So we've already looked at how the interaction between our EEG system and our MRI scanner can affect safety, and it can affect the data quality on both sides. So what you need to keep in mind is that compromise is often necessary if this technique is to be implemented successfully. And sometimes we get too enthusiastic about exploiting the potential advantages of this technique and forget that we might need to make some adjustments. So this is where we really advise that you think well ahead about your project, and you do some pilot testing, and you look at your data quality and you look at the safety aspects and decide where you can make the compromises to optimize your own experiment for what you want to get out of it. Okay, so what I'm just going to cover very briefly in the last part of my talk is EEG-fMRI applications. Like, there are so many of these, it would be impossible to cover all of them. But what I would point you to is a review article that I wrote that does cover these, and the reference is in the handout. So what we can think of really is where EEG-fMRI started. So the origins of using this technique, and pioneering those first developments were the epilepsy researchers, and also to some extent single trial ERP analysis. And these things paved the way for a lot more EEG-fMRI research, specifically in cognitive neuroscience. Now, there are so many cognitive neuroscience applications, I couldn't list them all on one slide, but there are many applications where EEG-fMRI can be very helpful in terms of providing this complementary spatial and temporal resolution. Sleep is also an area of research that has benefited hugely from EEG-fMRI. And then, as with all techniques, once it becomes more established and well used, we want to take it even further. So the developments in how to apply EEG-fMRI are still evolving. So we have a number of emerging techniques that are kind of on our radar at the moment, at least. And we have the two extremes of ultra high field MRI. So there are 7 Tesla, for example, is becoming far more commonplace. And then we also have the other extreme of ultra low field. So, for example, in the newly developed scanners that can do MRI at the bedside, combining EEG in those kind of applications could also be interesting. Now, brain stimulation is generally a hot topic in cognitive neuroscience and clinical applications. And combining that with EEG-fMRI is becoming more popular as analysis techniques develop. So that's an interesting topic to keep an eye on if you're interested in that. And neurofeedback as well. Now, neurofeedback is not new in itself, but combining EEG and fMRI, for example, to look at depressive disorder, has been quite fruitful for some groups. And then we also have the progress into trimodal imaging. So if EEG and fMRI is working very well, why not add something else? And in this case, what we're talking about is PET, so positron emission tomography, where we can look at some metabolic aspects in addition to the EEG fMRI. Now, I'm not going to go through examples of all of these. It is such a huge literature that we could literally spend an entire webinar on just this. So what I am going to do is just take a couple of examples to show you how EEG-fMRI has been applied and how it's then progressed from there. So as I said, EEG-fMRI has its origins in epilepsy research. Localization of epileptic generators is critical to understanding and treating epilepsy. And so EEG-fMRI has been crucial because the epileptic activity does generate some changes in the EEG. So we can pick up interictal discharges or spikes using our scalp EEG. These discharges also cause an increase in metabolism and blood flow that we can measure with the bulb signal. And so combining the EEG-fMRI is really almost a no-brainer in this respect. So if your patient is in the scanner, you can't always see if they're having some kind of seizure or an epileptic discharge. But if you see it in the EEG, you can then use that time course to model your fMRI analysis and reveal which brain regions are associated with that response, but also more importantly, how that propagates, so how it affects areas that are not necessarily generating that response. So this is a really nice example of where EEG and fMRI are truly complementary. The other example that I want to show you is single trial analysis of the ERPs, and this is something you're going to look at a little bit later. So what we can do is add a regressor representing the trial-to-trial variability to the MRI analysis to see which cortical regions are involved. And the example here from way back in 2005 is looking at single trial error-related negativity. So this reflects adjustments in cognitive performance monitoring. And by including this trial-to-trial measure from the EEG into the MRI analysis, you can highlight brain regions that play a key role in this processing. So this really shows how the trial-to-trial EEG-fMRI response is actually linked to behavioral responses as well. So this is a really nice application, and this did open the door for many further applications. Okay, so to wrap up, EEG-fMRI has become a very well-established research tool, and with careful planning, the challenges associated with EEG-fMRI can be easily overcome, and it does allow us to measure complementary signals concurrently. And many research fields have benefited from EEG-fMRI. So it's advanced our knowledge of basic brain function, so we can investigate that process of neuronal and vascular responses, so neurovascular coupling, using EEG-fMRI. We have investigated complex cognitive processes in the brain at rest, and we've also managed to push the investigation of treatment methods, such as in epilepsy or depressive disorders. So EEG-fMRI has really contributed to the science, and is a measure that is just waiting for you guys to use it. So I would switch to questions right now. We do still have 5 minutes or so until we have the break. So Celia has been checking out all of your questions for me, and hopefully you have some nice ones. Yeah, so Celia, do we have any questions?
Yes, there's one question from the audience. So
Yep.
you mentioned that separate sessions might be better if artifacts cannot be corrected or not acceptable. How do you decide what is acceptable?
Yeah, good question, and especially if you're new to this, that can be quite tricky. So we generally recommend that you do this in a process of pilot testing. So for the EEG, you need to make sure you can find your signal of interest. So we recommend that you measure your EEG outside the scanner and inside the scanner and process it in an appropriate way and compare the data to see if you can see the responses both inside and outside of the scanner. And you can also do that in a step-by-step approach. So you can measure it in the static field, then you can measure it with the gradients running, for example, and you can see the additive effects of each step and how that affects your signal of interest. So really, yeah, for the EEG, yeah, it's really just, can you find what you're looking for? And you will pretty soon come to a conclusion about whether it's acceptable or not. In terms of the MRI, it's more about looking at your images. So as I showed you, you can see the electrodes and you do get some signal dropout. So we do recommend that you take a look at your B1 field and your B0 field. So any susceptibility artifacts you see or anything that might influence your B1 field. Just eyeballing the data, you can get a good idea about whether it's appropriate or not. And then we do also recommend a pilot test of running through your analysis. So run your fMRI analysis as you would want to, especially if you include any segmentation techniques in there. And that should then give you a pretty clear indication of whether you expect to see or whether you see the changes you expect. And again, for a comparison, you should be doing your fMRI with and without the EEG cap. So you can then see if the EEG cap presence has a detrimental effect. So, and again, that's not for every subject, it's just for your pilot testing. Okay.
Anything else? Thank you. Maybe I'll ask one more question before we take a break. And this time ask about maybe some of the experimental setup, specifically what would need to be adapted in the paradigm for an EEG-fMRI experiment when you're coming from an EEG background.
Yeah, okay, good. Yeah, I touched on that a little bit, but I didn't give too many details. So it depends a little bit on whether you're looking at an event related or a block design. But in both aspects, you should think about the timing of your stimulus or your blocks. So as I mentioned during the talk in EEG, you can generally have a faster presentation of your stimuli, but there are ways around that in MRI in terms of how to optimally plan an fMRI task. And there are some tools available, I believe, to help you with that. So your kind of interstimulus interval is something that you should consider. Something else that you should consider is that especially if you were just planning to move an EEG experiment straight into the MRI, you need to make sure that your stimuli are not time locked to your MRI acquisition, because we use a template subtraction for the artifacts. So if your stimulus is always occurring at the same point in a volume, then it's likely that you will reject your task response along with your MRI artifacts. So it's mostly about the timing of the paradigm and whether it will work in both, and making sure it's not time locked to your MR acquisition. Okay.
Nice. And I think we're right at like 13 now, or 2 p.m.
Great.
So
Yeah,
We
so
can take a break.
yeah, we'll take a 15-minute break. We will